Hello, everyone. This is going to be a short talk about our current project, which deals with splicing-derived neo-epitopes in pediatric high-grade glioma as targets for immunotherapy. So currently, there are two major types of immunotherapies: immune checkpoint blockade inhibitors and adoptive cell therapy, which mainly includes TCR and CAR T. And although cellular immunotherapies have had tremendous success, there are still certain limitations, particularly in the case of childhood cancers, where there is a low mutational burden, which renders them invisible to adaptive immune response. And in case of CD19 and CD22 CAR T therapies, they target the B cell lineage markers. So, therefore, these approaches are not scalable to other hematopoietic and solid tumors. And for the current targets of CAR Ts in both adult and pediatric high grade glioma, there are certain limitations like T cell intrinsic issues of poor trafficking, low persistence. Then there are always antigen centric issues like heterogeneous pattern of expression or loss of antigen. So there's always a need for novel source of tumor antigens to expand the scope of current therapies. And one way to get there would be through alternative splicing, which from our old work as well as many others have shown could be a mechanism for epitope loss and chemoresistance. But our more recent work of CD22 in BLL shows that it could be also a rich source of new epitopes in tumors. So we wanted to extend this pipeline to in the realm of solid tumors, particularly high-grade glioma. So for this, short read sequencing was done on 184 samples of high-grade glioma and 7 normal brain samples. And RMAT splicing algorithm resulted in 1,288 comparisons. Now, HGG-specific splice variants were further filtered for single exon events and delta psi to be of zero point 0.5. And these recurring events were further filtered to be in frame and localized on the surface, and we even filtered them for very high expression. So our analysis yielded a list of around 50 or so ecto-LSVs. And here I'm showing you the example of few of our targets. Now, the plus delta psi indicates that these exons were skipped, minus indicates that these exons were included. And as you can see, the most important thing what we noticed was for most of our targets, the length of the exons was very small, which corresponded to being them called them as microexons, which have been extensively described in neurons in brain. And these encode for microdomains, which are important for protein-protein interaction, and their dysregulation has been shown in autism and many other brain-related disorders. So coming back to NRCam, which is one of our favorite targets, it shows the skipping of two microexons, exon 9 and exon 23. And as you can see from the splice graphs in green, the normal shows that majority of the normals include exon 9 and 23, while the more than 80% of the high-grade glioma samples show a skipping of these particular exons. And if we look at the junction, which corresponds to the skipping of exon 9 and exon 23 in high-grade glioma, which is shown with the green bars, almost none of the normal tissues of GTEx express this junction, and there are some neural tissues which did express, but still it suggests that these junctions might be specific to high-grade glioma. So we wanted to know whether the skipping of exon 9 and exon 23 are happening in the same transcript. So for that, we did long-read sequencing for an RCAM in KNS42, which is a high-grade glioma cell line, and a PDX3058. And you can see the reads, almost all of them suggest a complete skipping of exon 9 and 23 in the same transcript. So this is consistent with the dominant transcript being 351. 718.8, and according to Xena portal, this isoform is expressed more in HGG, which is shown in teal, compared to normal brain shown in purple, which is also consistent with our data. So now that we have identified our transcript of interest, we wanted to know how is the expression pattern at single cell level in the tumors. So for that, we collaborated with Dr. Tilgner, who have developed the technique of single nuclei isoform RNA sequencing. And we used 3058 for our pilot test. And here on the right, I'm showing you the data from Sizerwiz plots, which are generated from the long read part of the pipeline. And what you can see is the reads that span from exon 8 to exon 10 show a complete skipping of exon 9 in almost all of the cells from all clusters. And I'm taking another example, CHL1, where we expected the microexon to be included. And you can see again, read spanning and adjacent exons show a complete inclusion of these particular microexon. And this is actually in stark contrast with the cassette exon, which is exon 33, where you can see a more variable pattern of inclusion and skipping. So suggesting that our targets show a more less heterogeneity and more homogeneous expression, but we are wanting to expand this analysis to more PDXs and primary tumors to confirm our observations. And the second question we had was, so do these splice variants, do they make it to the cell surface? So using our KNS42 cell line model and bioattenuation assay, which we do show that these NRCam skipped isoform makes it to the cell surface. And do they contribute to gliomagenesis in any which way? So for that, I went back to my model of KNS42, where we created knockout for NRCam, and I re-expressed the canonical and the skipped isoform of NRCam. We did not see any changes in proliferation of these cells, but we did find that the expression of the skipped isoform of NRCam enhances the migration and invasion capacity of high-grade glioma cells as compared to the canonical isoform and the knockout cells, suggesting they do have a role in glioma progression. So now when we have addressed all these questions, our current goal is to develop binders to NRCam skipped isoform. And for that, we collaborated with Fred Hutch Antibody Services, which uses the whole cell immunization approach, where BALP-C mice are injected with NIH3T3 cells, which are overexpressing my isoform of interest. This is followed by the production of hybridomas and then flow cytometric screening for the binders. So from this pipeline, currently we have a binder 3F8, and which shows selective binding to the skipped isoform of NRCam, which is shown in red, as compared to the full-length NRCam, which is shown in green. And this data is generated in CHO cells, which are overexpressing these different isoforms. We also have data with KNS42, which endogenously expresses the skipped isoform. And you can see that 3F8 nicely recognizes this endogenous isoform compared to the knockout cells. So currently, we are expanding our pipeline to many other targets beyond NRCam to determine their contribution to gliomagenesis. And most importantly, we want to develop these antibodies and test their specificity against panel of HG and pediatric tissue microarrays. And currently, we are in the pursuit of making some of our favorite immunotherapeutics into ADCs and CAR-Ts and test them rapidly in vitro, in vivo, and against a panel of HGG PDXs. So with this, I would like to thank my lab, especially for Andre to let me work on this project, and my teammates and our collaborators who have ensured that this project moves at a great rate. Thank you so much.
Thank you so much, Priyanka. Any questions?
Great talk.
Thank you.
I'd like to ask a question that sort of would bother me if I had to approve this further, if I was working for the FDA or something. So CAR T cell therapy has problems with toxicity and with escape. Do you think that these types of mechanism, alternative splicing and such, are like hard switches on and off? They'll have few toxicity and no escape? Or
okay,
how do you
so
see that?
I'm going to answer in two parts your question. So your question is about the toxicity. So off-target toxicity, we are trying to avoid by ensuring that our target is as good as it can get. I can give you an example of the current target, which is being pursued in the clinic.
My question is really much more general. So what I'm worried about is that these alternative splicing events are not a hard switch.
So
they're always moving and they're not stable.
I have heard this question before, and I went into EGF variant 3, which is known to skip around 2 to 7 exons from 2 to 7. And I found consistently that variant consistently changed, and even with immunotherapy, it actually never reappeared, or the cells never actually showed a pattern of escape. What actually happened was the adaptive immune response showed increase in PD-L1 and other stuff. So the compensation happened at a different level, but the isoform continued to show stability throughout the treatment and otherwise. And one way to ensure that these antigens don't escape was to make sure that they have a certain role in the progression of the tumor, because if they're important to the tumor won't want to get rid of it. And that is why we are trying to find if functionally they are helping the tumor in any which way. And that's what we hope, that it won't escape.
Very nice talk. Do you have an idea how these two exons are regulated? And do you see any misregulation of splicing factors in high-grade glioma?
I am so happy you asked this question. For that, I have a slide for you, and I'll quickly flash it. So actually, microexons are known. There are some traditional splice factors which are known to exist in neurons and astrocytes, which can cause splicing changes. And when we look at the expression of these splicing factors, I do see that they are differentially expressed, like HGG shows lower of RBFOX1 as RRM4. We actually did analysis where we looked at especially the skipping of these two exons with splice factor. There are few that came up, and I tried to overexpress them. I found to some degree the inclusion happened, but it was never 100%, even though I was adding selection markers and everything. So I believe there may be multiple factors, and if I try to express them together, I might see changes. But yes, we did look into this analysis a little bit. So we have some idea of the RBPs that may be playing a role here.
Thanks.
Thank you.